Hi class, in this lecture we are going to be looking at genetics. So let us get started with what we are looking at today. The first thing we are going to look at is what is genetics. Genetics is the study of heredity. We'll look at the structure of the bacterial chromosome. We'll talk about what genes are and what make up the genes. We'll also discuss genotype and phenotype. We'll also talk about the sizes of them. That is what we are going to look at today. Alright, so again, I talked about the genetics, the study of heredity. Genetics explores the transmission of the biological traits from one generation to the next generation. This is the essence of what genetics are. Genetics explore the expression and variation of the traits. Genetics explores the structure and the function of genetic materials. Alright, so again, just knowing some of the basics. The genome is the sum of genetic material. And again, this can be chromosomes, mitochondria, and chloroplasts, and or plasmids. When we talk about cells, the genome is DNA, while the genome of the viruses can be either DNA or RNA. Alright, and then when we think of DNA, it's complex with proteins, and this makes up the genetic materials to form chromosomes. Okay, we look at bacterial chromosomes. Bacterial chromosomes are a single circular loop. Thus, if we look at the prokaryote, we can see a single loop of double-stranded DNA, roughly between 4 to 5 million base pairs. When we look at eukaryotic cells, these tend to be linear pieces of DNA. Now, it does not seem like they are linear because it seems that they are really compacted in it. But these are linear pieces, so they have a definite two ends to each chromosome. Each chromosome contains many genes, and the average eukaryote chromosomes tend to be about 100 million base pairs. Viruses have a minimal genome. Viruses only have a couple of genes in order to survive, and typically their DNA or RNA is from about 100 base pairs to a couple of thousand base pairs. Alright, we look at the chromosome. A chromosome is divided into genes, and these are the fundamental units of heredity responsible for a given trait. Genes is the site on the chromosome that provides the information for the cell function. Gene is the segment of DNA that contains a necessary code to make the RNA, which will then make the protein. And so this is what we talk about when we look at protein synthesis. So the DNA is the blueprint, the RNA is the messenger, and the protein is the product. And again, we have three different types of genes. The first type in the majority of the genes is going to be for proteins. We call them structural genes. There is another set of genes that are the code for RNA. This RNA is either for the mRNA or ribosomal RNA. And finally, gene expression. These are regulatory genes, which will turn things on and turn things off. These are going to be important when we talk about operons. Even though we are talking about bacteria here, we need to know a little bit about genotypes and phenotypes. Genotypes refers to the organization of the gene. All types of genes constitute the genetic makeup. Phenotype refers to the observable traits created by the expression of the genotype. What does the gene to produce? What does the trait looks like in the organism? Here is the eukaryotic organism. This is a plant. When we refer to phenotype and genotype, the genotype refers to dominant and recessive. In bacteria, we do not have dominant or recessive, only one genotype for each phenotype. So again, when we talk about plants, we talk about petal color. In this case, the phenotype of purple, the genotype would be dominant versus dominant or dominant versus recessive. The genotype of recessive versus recessive would give you white. However, in bacteria, we do not even have to worry about that because they only have one genotype. Alright, how are the sizes? The smallest is the virus that has roughly 4 to 5 genes. Some can be more complex. This comes out about hundreds of base pairs to about a couple thousand base pairs. E. coli has about 4,000 genes, and this is equivalent to about 4 million to 5 million base pairs. If you pull it out of the cell, it will range to about 1 millimeter in length. So if you look at your millimeter scale, that is how big the E. coli genome is. I like this picture here that shows you the E. coli is releasing all its DNA out. You can see how compact the DNA gets put into the bacterial cell. If we compare that to ourselves, we have 46 chromosomes compared to 1, and we have about 30,000 genes compared to 4,000. So we have much more DNA packed into our cells. If you took all the DNA out of one chick cell, it will range to about 6 feet in length. So if you are under 6 feet tall, your DNA would be taller than you. It is pretty impressive by thinking of you look at one chick cell, its nucleus has 6 feet of DNA packed inside that cell.